Hi everyone, and welcome back to my channel. My name is Chacha, and today I'm going to be talking about neurons and action potentials as well. Before I get into the video, I just want to say that this channel is created just to help me study for the MCAT and hold myself accountable. And I am a student myself, so I could make mistakes. And I apologize in advance. And please tell me in the comments if I made any like super big ones. So before we kind of jump in, like we should probably talk about neuron anatomy. And really like the only different like there's multiple different types of neurons. We're just like gonna be primarily talking about the ones in like the CNS. There's also like cardiac, like when we describe like action potentials and stuff like that, like cardiac cells are important too, but they're not really stressed on the MCAT. Um, yeah, we're talking about unicellular neurons, and unidirectional. So here we have the dendrites. This is like the labeled one. Then we have the soma, which is two. The axon hillock, which is that light green color. The axon, which is that green. And then the nodes of Ranvier. I don't know how I'm saying that, but that's kind of in the axon, which I have here. They're just like pretend. They're like the places that aren't myelinated. And then over here in the light blue color, which I'll label as 5, that's where you have the axon terminal, which is what in the presynaptic cell, which will meet with the dendrites of the postsynaptic cell. Okay, let's kind of get into more of the nitty-gritty stuff. So dendrites are what accepts neurotransmitters from the presynaptic cell, which is what I said earlier. And typically they have like large surface areas. They're made of actin and microfilaments. And they're pretty dynamic, so they're always moving. As I talked about in a prior video about like long-term potentiation, right? So it really like depends, and also just pruning in general. So like if that neural connection is strong, then obviously like you're gonna want to have like more surface area. There's gonna be like a lot more property, supply and demand, right? So for example, like if you have like a nice like beach house like strip like in Florida or something like that, right? Like you're gonna want to have like a lot of houses built on it. Then like if you're in the middle of like I don't know, Nebraska. Like no one, I'm just no hate to Nebraska, but there's it's probably not as high in demand. That's kind of the same thing with dendrites. The soma, that's just kind of like where all the nuclei of the neuron is. So like just like all that fun stuff. So like cytoplasm, so it's like the Golgi is there as well. So just kind of like the like organelles are in the soma. Then the axon hillock, I like to kind of think of that as the accountant. So it's like kind of like right before where the axon is, and then it's kind of like tallies up like all the excitatory and inhibitory signals, and it's like it gives like the A-OK if like let's say like oh you could pass go. So like you if you pass go, if you pass that threshold, then you're gonna fire an action potential. But action potentials are all or none. So typically like when like your teacher or someone will explain what the threshold of an action potential is, I've always learned that it's negative 55 millivolts, but sometimes people have like different perceptions. Oh, sorry. Different perceptions. But so if you have like negative 54 millivolts, it's not gonna fire. It's like really, really strict. It's kind of like a bouncer. It's like it's strict. You either have to be like if you're like 21, right? Or it's like if you're 20 in like 364 days, that bouncer's not gonna let you in. But if you're 21, even if you're your 21st birthday, right? They'll let you in. That's kind of what an axon hillock is. So it tallies up everything, make sure everything's checked, the checks and balances, everything's in order. Um, an axon, which is like here, that's in the darker green color. It's where the action potential it travels down to the axon terminal. So typically like axons are just like having it like travel like wide, not necessarily like super long distances, except for there are like definitely cases, especially in the PNS or your peripheral nervous system, where axons can be really long. Um, but it's just like kind of like a long space for the action potential. So like for those different cell types to communicate as efficiently as possible. And then it could be myelinated, which is like when you see like white matter in your brain, or it could be unmyelinated, which is gray matter of your brain. So white matter is typically fast. Myelination is kind of like you could think about it as like insulation. So it's keeping that electrical signal localized. So whereas so like if you don't have the insulation, it can kind of go like all over the place. Like it's not in like the electrical signal is like not in like one place, hence the localization. It's like it's really hard to keep the action potential from going the full distance. And it takes more time, which hence gray matter, which is slow. Typically it's like for like processing information. Whereas white matter is like fast. It's crucial for like transmitting information like as fast as possible from cell to cell. So like if there's something really important, you do white matter. If something's kind of like slow or you're like kind of thinking about it, it's gray matter. That's just a general rule of thumb. Typically, like you won't be asked about that on MCAT though. And then I also want to talk about the nose of Ranvier, which is like what I crudely draw, like drew here. With that's not what they look like at all. They're like gaps between the myelin, but I don't really know how to draw, hence my really poorly image, as you guys can clearly see. Um, but think node, like back into physics concepts, like when you're talking about waves, right? So like a node is like where it's zero. So it's like it's that zero value. So that means that like there's no myelin there. And like why are the nodes around here? Like what's the point of having like no myelination along the axon when we literally just talked about how myelin is really important for insulation and propagating signals. It's important for saltatory. Oh my God, I can't. So yeah, I think that's how you say it. Saltatory conduction. In Latin, saltus means to leap. So it's like when your action potential is jumping from one node to the next. And why is it important to kind of like keep like the action potentials kind of separate from one another? So like think about it. We'll get more into like the graphs of action potentials. Think about it. You have like sodium and potassium just all around, and it's like you have like those voltage gated um like ion channels, right? And it's really important, like when you have something that's voltage gated, that it reaches that voltage. But if you have everything all like happening all at once, the signals can get muddied, and it might not like there might be some overlap, and you may not necessarily reach that signal. So they're less likely to have an action potential occurring. It's kind of like there's like too many cooks in the kitchen, right? Like everything just guys kind of goes to crap. Um, and then in the light blue, I have my axon terminal over here with five crudely drawn. And then the axon terminal, like what's it's really important for is exocytosis of the vesicles. And your vesicles are released by calcium. So it allows calcium into the not in the synapse. Oh, sorry, it releases neurotransmitters in the synapse. Sorry, I thought I wrote something that wrong. It allows calcium into the presynaptic cell in the axon terminal. So here I kind of drew just like a wider picture of it. So this is the light blue. It's a presynaptic cell. So VG, voltage gated calcium channel. So it allows for voltage influx into the presynaptic axon terminal. And then calcium, it can bind to a receptor on the vesicle that holds like neurotransmitters. So like in the presynaptic cell, neurotransmitters are typically held in vesicles. And then once that calcium binds to said neurotransmitter, it'll like it starts a signaling cascade that essentially has the vesicle go towards the membrane, and then exocytose, which is literally like think about it like vomiting the neurotransmitter out into the synapse. And then the neurotransmitter that I drew here, it can bind to a receptor, and that receptor can can be like a signaling cascade in this postsynaptic cell. It helps like depolarize that cell. So like if a lot of the times it'll be sodium or calcium. Like sodium is for like AMPA receptors, which I talked about in long-term potentiation, which for glutamate. And then another one like calcium, for example, it's for NMDA, which is calcium. Yeah, sorry, I just said calcium, but that's also for glutamate. I apologize. Yeah, and then it can just keep propagating the signal until where it needs to go. Okay. Before I kind of get like further into action potentials, I want to talk about the different types of glial cells. Glial cells, I kind of think about them like of the support of the cell. So you could think of like the neuron, they're kind of like the star of the show. But like let's say like they're like a pop singer, right? But it's like even pop singers, even Taylor Swift, like she has like a crew that makes her able to perform as well as she does. And she has like a whole team behind her, and they're just as important. In the PNS, there's like really like one type of specific glial cell that I want to cover. Everything else in the PNS, hence like it being in the periphery, um, excuse me, being in the periphery, it has like all the other types of cells that you'll see like outside of the central nervous system, including like the immune cells and just like stuff like that. So Schwann cells, it's really distinct between like oligodendrocytes is that one Schwann cell per neuron slash axon is what myelinates it. So they're really important for myelination. Um, and also I just say the CNS too, that's like your brain and your spinal cord. That's everything that's in considered the CNS, our central nervous system. So central, it's like central in your body, like if you were to think about it, right? Or centrally, you could think that it's like what's like most important for your body. It's the center of command. And then, as I said, oligodendrocytes are what myelinate multiple axons of neurons. Then you have your microglia, which are your immune cells. They're phagocytes. So phagocytes are like literally like they surround what they like mark as like something other or something that shouldn't be there, and they just kind of like eat it. This is my phagocyte. Boom. It absorbs it. Yeah. Into itself. They're kind of like the trash cans. They're a type of macrophage, which are phagocytes. And then astrocytes are what maintain the blood-brain barrier, or BBB. Um, this is also kind of like another type of bouncer of the brain. So it's like it's really important to maintain the integrity of the CNS, just because like there's all sorts of toxins and viruses and all that stuff that can get into your brain, and they could be really harmful. For example, like if you have like encephalitis, which is like inflammation of your brain, that can be deadly. And as I said, as I've mentioned in my prior videos, like you only have like a set number of neurons. You're actually pruning a lot of your neurons as you age, just to like maintain efficiency. The only place where you're actually making new neurons is in your hippocampus. Everywhere else, it's just kind of like you only have like a finite number. So it's really important to maintain that. And if like something were to get into your brain that would damage your neurons and or cause them to die or something like that, that's really, really harmful. Your brain is just like so efficient, and just like a couple of neuron loss could be detrimental. Um, then I want to kind of, before I go into like action potentials, like specifically, I want to talk about like temporal summation and spatial summation. Oh my gosh, sorry. My voice is like kind of going away. Give me 1 second. So, temporal summation is like the same presynaptic neuron. It sends signals, vesicles, like it releases like vesicles of like neurotransmitters to the postsynaptic neuron in quick succession. It's perceived typically as one signal. So basically it's just like kind of like massive depolarization events. That's how what I like to think of temporal and temporal like tempo time, so like that makes sense, right? So it's like just like massive succession, it's like ba boom ba boom ba boom. Whereas spatial summation is when multiple presynaptic neurons send signals and release vesicles to the postsynaptic neuron. So this can also result in a massive depolarization event, or it can also be like just I guess like temporal summation, or it could also be sorry, an inhibitory event. So it can be send inhibitory signals, or the signals can cancel out since different neurons can send different signals. So you could totally have a cell, like let's say like this green one, this green presynaptic neuron, that's sending an excitatory signal, but this blue one is sending a inhibitory signal. So it's very likely that they would cancel out, or maybe one is like slightly stronger than the other, but as long as it doesn't reach that -55 threshold, the axon hillock, like it'll be like, no, like you can't send out an action potential. Okay, what the heck is an action potential? I feel like we've been talking about this the whole video, and now let's finally get to it. Um, so first things first, I kind of want to talk about like the different like I guess like what's on the x-axis versus y-axis. So time, typically their action potentials are electrical signals, and if you think about lights turning on in your kitchen or bedroom, right? Like when you flip that switch, it's like a matter of like milliseconds. Same thing with action potentials. Whereas on the y-axis, it's in millivolts. Um, at negative, I want to point out like the different numbers that are on the y-axis. So you have -70, -90, -55, and +30. So -70 is like the resting membrane potential. So that's just what your neuron is at rest. Like there's nothing going into it. -55 is that threshold we were talking about with the axon hillock. It's the all or none, so you must reach this value for an action potential to occur. Um, for those voltage gated sodium channels to open. Um, whereas depolarization, it's from -55 to +30, and that's when those sodium channels open. So in your cell too, like if you have inside, oh, I should just do in, out. So inside you have a high concentration of potassium, outside sodium, and typically chlorine. And you have various negative in here. Um, yeah, so just like kind of thinking in your mind. So like if you remember, right, like just like for diffusion, right? When you have like those channels open, like it wants to go from high to low, which is the same thing. So if you have opening of voltage gated potassium channels, um also another thing is that this is like higher, it's positively charged out here, whereas inside the cell it's negatively charged, right? So the concentration and charge differential is what drives sodium to go inside the cell once those voltage gated channels are open. So it doesn't require ATP. And then they close at +30. So like that's when there's an inactivation gate. So that's like think about an inactivation gate as being like the upper limit. Whereas for repolarization, potassium is like when they by the time it takes to get to 30 millivolts, like with those Ne plus concentrated sorry ions going into the cell, that's like when you get like potassium getting open. And also potassium is like extra like wants to go like leave, right? Because like now you have like all those positively charged sodium cells going inside the cell and sorry, ions going inside the cell. So when you have like too much of a positive charge, right? Like since like in electromagnetism, when you have two positive charges, they repel each other, whereas like sorry, opposites attract. So when you have like a lot of like positively charged ions in this very close space, then that kind of drives, it's like further drive of potassium to get out. Because now also inside of the cell is more positively charged than outside the cell. And one tad bit about potassium gated channels is that they close super slowly, which is what leads to hyperpolarization, which is like that -90 typically for that millivolt. So hyperpolarization, there's two different types that I want to talk about. So there's absolute, which is when there's no action potential at all, and then those sodium channels are fully inactivated. So if you have no sodium channels that are inactivated, then you can't have any depolarization, right? So that's what we said is like the main step after you reach that threshold. Whereas relative is like when you need a much higher excitatory signal. So like let's say like you needed it to be -55 is like typical. Let's say you need it to reach -40 instead. So there's like a partial recovery of those sodium channels during that time. Okay, so we were talking about that, right? But like How does it go from -90 over here to -70, the resting membrane potential, if there's no more of those voltage gated channels that are like in play? So one of the things that's always open is like leaky potassium channels. Um, and that helps maintain the negative charge, which is like not super relevant in this case. That's just like maintaining after it already reaches that -70. But the main one is the sodium potassium pump, which is an ATPase. Which is like your favorite friend. This isn't just relevant in the neurons. There's a lot of places where you will see a sodium potassium channel. So definitely keep this in mind. So it goes against the gradient. So you're having 3 sodium out and 2 potassiums that go inside the cell. So if you think about it, if you're like always like kind of confused about, hey, like I can never remember, is it 3 sodium, is it or is it 2? And does sodium go inside or does it go out? Just like think about it in terms, excuse me, of like our friend like inside and outside. So like there's high, so like inside of the cell that it wants to be negatively charged, right? So that makes sense, right? You are going to want to have like more positive charge leaving than coming into the cell to maintain that negative charge. Okay, so that makes sense. Now, if because you know that sodium potassium goes against the gradient, which means that you're going from low to high concentration, it makes sense, right? That you're going to want to have potassium go inside the cell. Okay, so that makes sense. So when you put 2 and 2 together, you know that potassium wants to go inside the cell and that potassium in that inside the cell it's like negatively charged, so that means that you're going to want to have less positive um like ions go into the cell. That makes sense that it's 2K plus in, where it'll be 3Na plus out because you want the outside of the cell to be more positively charged, and that that sodium has a higher concentration outside the cell anyways. Um, yeah, so it's kind of how I like to think about it. So, like everything that goes against a gradient, you're gonna need some form of energy because the body just doesn't like to do that. Hence why you use ATP, and it's called an ATPase. Um, so here's kind of like my drawings of the ATPase or sodium potassium pump. So you have your extracellular matrix and your intercellular matrix. So first I have 3 Na plate, Na plate, Na Na plus. Oh my God, ions. So they're gonna go and they're gonna find their way into the ATPase and they find like where they can bind. Okay, cool. So then ATP, like through hydrolysis, that phosphate it'll go onto the oh my God, ATPase, and then this will spark a conformational change. So what does this conformational change do? Originally, the ATPase was facing the intracellular matrix. Now it's going to face the extracellular matrix. And at the same time, those sodium, like they'll release, so they'll go off into the sunset, into the extracellular matrix. And now, so that ADP will now become ATP again. And first, I guess I should say, like when it's still facing the outside, the sodium here, I'm not sodium, sorry, potassium will bind into the ATPase. And then with that ATP like no longer being a phosphate, an organic phosphate onto the ATPase, what happens? The sodium, it goes faces the intercellular matrix again. And then sodium will like go off into the sunset, into the intercellular matrix. And slowly but surely, if you could picture like a ton of these doing, it's not just one sodium potassium pump. There's a ton of these at the same time that are doing this. So eventually it'll reach that -70 again. And then yeah, it just helps maintain the resting membrane potential as well throughout. So hopefully that made sense. That was kind of all I wanted to talk about for neurons. Hopefully I didn't make any egregious errors and that the video made sense. And sorry if my voice is a little weird. I'm kind of sick right now. But not sick enough to study for them MCAT. Yeah. But yeah, hopefully that made sense and good luck with your studies.